I've got two different overarching areas in which we can address quality. I'd like to start first by looking at what we know about AAV analytically and how that can help us go on to make some of the process development decisions that we have to make, especially as it relates to a quality by design approach. So I wanted to start first. I'll put Andrew in the hot seat first here. He actually penned a paper a couple of years ago in 2021 about the importance of analytical characterization as the first step in implementing a QBD approach to bolster the quality and process reproducibility for AAV. So I'd like to start, Andrew. We've had 2 years, right, since this paper was published, 2 years more of experience. I'd love to get your sense, and then Justin, we can come to you. How well do we understand AAV CQAs today? And, you know, where do you feel we've made the most considerable progress in terms of quality attributes? And, you know, in which do you think we'd have a longer way to go before we truly achieve well-characterized?
Okay, well, thank you for welcoming me, and hello to everyone watching the stream. So to jump right into it and think about quality attributes. So I wrote the paper in 2020, 2021. It was part of kind of my training because I moved over from antibody process development and set up an AAV viral vector process development group at Regeneron. So as part of my training, I read everything I could read about quality, and then I thought, well, I've done all this, I may as well share it. So I wrote a review article on it. So what do I think has progressed the most? I kind of feel that when I did my reading and I wrote that review article, I think the MT4, what was an appropriate level of MT4, I think there was still some discussion that there may be some positives to having empty vectors. For example, they could kind of act as decoys for the host immune system. Whereas I think since that time, especially there's been a number of patient deaths in studies linked to very high dosing. So with issues such as hepatotoxicity, thrombotic microangiopathy, and neurotoxicity, especially with the dorsal root ganglion. I think that with that, and with more discussion around the industry, and especially as people have developed more potency assays, we've seen that in a potency assay, often more empty particles noticeably reduce the potency in the assay. So I think that we're all pretty agreed that we want to minimize empty particles as much as we can. And the level in which we minimize to is open for discussion and probably dependent on dose level and route of administration. I think there's a much more of knowledge around that now.
Absolutely. Yeah. Go ahead.
And then thinking about what probably hasn't come along as far, it's probably residual DNA packaging in the AAV. So I think we've definitely learned that empty capsids are probably not empty. They probably have may have small amounts of DNA in them. But as to kind of what is okay in terms of residual DNA, is there a particular type of DNA that is worse? If it's plasmid DNA with high levels of CpG, is that worse because of the immune response? Or are we concerned more about oncogenic sequences from the host cell, the human DNA, if using HEC293? That's kind of, I think that's still, the knowledge is out on that field. And I also feel that we've learned a lot from mass spec approaches on deamidation, these kind of post-translation modifications, but I do think there's probably still more to learn in that field as well.
Absolutely. So your argument here would not be that we are a super well-characterized, our AAV vectors are not necessarily well-characterized quite yet today. Is that your overarching argument?
They're definitely getting there.
Yeah.
And we have 5 approved products in the US market anyway, so
absolutely.
They've been good enough for those products to be approved.
Yes.
But there's always work to be done.
There's work to be done.
That's the exciting thing about the AAV field. There's a lot of unknowns still to be found out.
Absolutely. Justin, I'd love to turn it over to you and hear your thoughts as well on what Andrew's talked about. I know full empty gets a lot of attention in this space when we talk about quality, but I'd be curious to understand your take as well, knowing that we have products on the market. You've been working in this space for quite some time as well. You know, how well do you think we understand our AAV CQAs today, and where do you see the greatest progress is being made and some of the the less progress that, right, we need to see more being made.
Thanks. I'd agree a lot with what Andrew said to date. I mean, I've been in the space for about 8 years now and seen quite a bit of maturity as we've gone through this. And particularly in the last several years, I think it's accelerated around the characterization of packaging within capsids, from empty capsids to whatever you would deem, partially filled capsids or whatever moniker you give to them. There's been quite a bit of work gone out there in the industry and with Regenx on these items. And I think Andrew hit on this well. At earlier stages, it wasn't as certain of the maybe detrimental, the safety impact of some of these impurities. I think the picture is becoming a little more clearer that moving to lower levels of those impurities is probably likely beneficial for patient success and outcomes with it. So I think a lot has gone into the ability to characterize those. And with that, I think Andrew hit on a few of them around mass spec around looking at capsid modifications, as well as, trying to really get at in those partially filled capsids, what is in there? Can you characterize, what extent can you characterize that DNA to? And I think there is, more to be written on that story there. And it may end up being route of administration specific as to how important those items are.